The Prostate Cancer Foundation has been funding research and patient treatments with immunotherapy for many years. This has been a very tough road, as everybody knows. The immunotherapies that have been so successful in melanoma, lung cancer, and other diseases have been very, very difficult to reproduce for prostate cancer. But fortunately, that's beginning to change. So I'm happy to introduce first two talks that will address the issue of CAR T cells for prostate cancer. The first is from Tanya Dorff, who will discuss the clinical development of a PSCA-targeted CAR T cell for advanced prostate cancer. Dr. Dorff is from the City of Hope. The second talk is from Dr. Naomi Haas from the University of Pennsylvania. Dr. Haas will present data from clinical trials on the co-expression of a novel TGF-beta receptor dominant negative to enhance PSMA chimeric antigen receptor therapy in the treatment of castration-resistant prostate cancer. Finally, a talk from Alejandro Sepulveda from Janssen Research and Development will present on a first-in-class shared neoantigen vaccine for the treatment of prostate cancer. We are very interested in these talks and hope that everybody will be encouraged by these advances in immunotherapy. Following these talks, there will be a live discussion.
On behalf of my collaborators who are pictured here, whose work I'm largely presenting today, I'd like to thank the Prostate Cancer Foundation not only for the invitation to present our clinical development of the PSCA-targeted CAR T cell for metastatic castrate-resistant prostate cancer, but also thank them for funding this study. Pictured now are my disclosures, including the disclosure for the CAR T license. Cellular immunotherapy has an established role in advanced prostate cancer since the approval of sipuleucel T back in 2010. Now, there was controversy since there was no immediate, obvious objective response that accompanied the survival advantage, but multiple subsequent papers have shown mechanistic support, and subsequent analyses have confirmed a survival benefit. You can see Rhonda McKay's real-world evidence analysis, and the PROCEED registry recently showed a specifically large benefit in African American men. However, we would all like to find immunotherapy that is a bit more potent. So that's where CAR T and BiTE antibody is being explored for prostate cancer. And you can see pictured some of the targets that are being used. But the first step when you have a targeted immunotherapy is to define the toxicity based on the normal tissue expression of these targets. And also, of course, look for a preliminary signal that this is a target that activates the immune system against the cancer. So at City of Hope, we have a broad cellular immunotherapy program in multiple solid tumors, and we're all working together to try to learn from each other's experiences, since we believe it will likely take more than just the CAR T to get us to durable, complete remissions, maybe combination therapy, maybe repeated dosing, and we have the benefit of learning from each other. Prostate stem cell antigen, PSCA, was selected as a target for our CAR T because it is widely expressed in prostate cancer tissue, even more so in metastases. There is some normal tissue expression, including the gastric lining and the bladder, but an upside of expression in the bladder and pancreas is that these cancers overexpress PSCA, and so the development of one CAR T could potentially be useful for 3 separate cancers. We don't yet know whether PSCA is an androgen-regulated gene, so that might impact timing of use of the product or optimal patient selection. The CAR T, the second generation CAR T that we're using, is a 4-1BB co-stimulated CAR T, and you can see in the bottom right graphic that this achieved greater complete responses in the mice and better survival compared to the second to lowest line with the CD28 co-stimulated CAR T. The phase 1 trial that we've designed starts with pre-screening patients using archival tissue, often their prostate biopsy or prostatectomy, to make sure their cancer expresses PSCA. After the leukapheresis, during the T cell production phase, we do an on-study biopsy. This is again used to confirm that the current cancer expresses PSCA, but more importantly, can be matched to the day 28 biopsy sample, so we can compare the tumor microenvironment and the immune milieu before and after the treatment. There is cytoreductive or lymph-eating chemotherapy being used for most cohorts, but you can see the BID cohort is where we started, 100 million CAR T without lymphodepletion. We then escalated to 100 million with lymphodepletion. We're currently expanding that with a goal of increasing to 300 and 600 million CAR T if we find adequate safety. The CAR T cells, we wanted to see if they could work by themselves, in part to help define the toxicity with this novel antigen. And the role of lymphodepletion in solid tumors has been questioned. However, as we can see here, that the CAR T by itself was not as successful at eliminating metastatic prostate cancer tumors in the mouse models, but the addition of cyclophosphamide made a significant difference. Because our full clinical data set is not ready for presentation, we're still actively accruing and treating patients. We decided for today to just present one patient's experience to give you a sense of efficacy, toxicity, and correlative science, how all of these are going to come together to help us develop this agent moving forward. So this patient is 73 years old, has had metastatic disease for many years, and is quite heavily pretreated, as you can see, with chemotherapy, pembrolizumab, even lutetium PSMA. As he was waiting enrollment on the CAR T trial, he developed a pancreatic tumor, but biopsy proved that this was metastatic prostate cancer and not a new cancer. You can see his on-study tissue staining with PSCA on the left there. He's the second box on the right-hand column of tissue. So as he was getting prepared to enter the study, his PSA was rising, as you can see in the graph. We did treat with cabazitaxel and carboplatin as a bridge with some control. Then we do monitor PSA after infusion of the cyclophosphamide and fludarabine lymphodepletion. You can see a drop in PSA there. Then there's a nice drop after the infusion of the CAR T, indicating some element of clinical response. This was matched by the bone scan findings with reduced uptake in the right humerus and left femur lesions. And in the CT scan images, you see less soft tissue around that metallic stent in the pancreas, so a reduction in the soft tissue tumor. His on-study biopsy was of the right iliac bone. You can see strong PSCA expression on the left. The post-treatment biopsy from the same area showed much less cancer, which was gratifying, but still preservation of PSCA expression, which is important because we think PSCA, loss of PSCA expression could be a potential mechanism of resistance. One of our correlates is to try to better define responses in bone, because this has been challenging, as all of you are well aware, in the prostate cancer world. We'd like to have objective measures and not just delay in time to cancer progression on bone scans. So my colleague Amar Chowdhury from radiology has found that there are some changes in the MRI that could potentially be used to quantify response. So you'll see the pretreatment scan on the left. There's peripheral enhancement and T2 hyperintensive signal in the right iliac bone met that is reduced or eliminated on the right-hand panel. Additionally, you can actually see decrease in the tumor size in that right iliac bone from the left upper panel to the right upper panel. And when you compare that to the CT findings, that sclerotic lesion, if anything, maybe looks a little bit bigger, a little denser. That can sometimes be mistakenly read as progression, but of course we know it could represent healing. But again, the potential for an objective, quantifiable measure of bone response would be very helpful. Another part of our correlative science is a collaboration with Peter Kuhn at USC, and I'd like to specifically thank Aidan Moriarty, a PhD who works with him, for the next 3 slides showing this patient's peripheral blood and bone marrow circulating tumor cell. This is actually using their single-cell liquid biopsy platform, which does not select for cells by phenotype. So we're not only looking at classic circulating tumor cells, but a whole host of rare cell events, which is a great platform for discovery in this trial. When we get bone biopsies, we are trying to get marrow samples as well. And this has been very important. As you can see, the cell populations in the peripheral blood on the left are very different from what we're finding in the marrow on the right. And often we find 1,000-fold greater number of cells in the marrow. The dark red represents the cytokeratin staining cells, which are the classic CTCs, which are not so common in the peripheral blood in this patient, but quite common in the marrow. And are reduced after CAR T treatment that correlates with his clinical response. The Vimentin positive cells, these teal-colored cells, expanded in the marrow after treatment, but were significantly reduced in the peripheral blood. So we have a lot to learn about what these various cell populations mean, but the overall picture is that there's a decrease in these circulating tumor cell or rare cell events in both the peripheral blood and the marrow for this patient after his treatment. Looking at the landscape of the cells that we see, the marrow, again, shows those red or almost pink cytokeratin staining classic CTCs, and on the bottom panel, those are almost completely gone. You see a few of them over on the right upper panel from the pretreatment peripheral blood, but not nearly as many. And taking a closer look, on the upper left, you see that classic CTC with a large nucleus and the cytokeratin red staining. Just below that, you see a cluster with multiple CTCs, as well as an immune cell and a Vimentin positive cell interacting. This looks almost like a tumor microembolus. With Peter Kuhn and his lab, we're hoping to develop the ability to look for the CAR T and how it is interacting with the other cells in this platform. Also, we're developing staining for PSCA so we can real-time look for PSCA staining cells and how those change after treatment. So we've seen a little bit about the effect in this patient. What about the toxicity? He did have cytokine release syndrome, which is a classic toxicity of CAR T therapy. It started around day 4 and was primarily fever and nausea and vomiting. The tachycardia did flip him into AFib, but he was never hemodynamically compromised. Day 6, we finally offered tocilizumab just because the ongoing fevers were so exhausting. So this was not dose limiting, but unfortunately, he subsequently developed hemorrhagic cystitis. This was initially dysuria in frequency, but culminated with hematuria necessitating blood transfusion. So this is something that we think could be an on-target, off-tumor toxicity. You can see in the upper right the radiographic manifestation of the cystitis. On the bottom right, his stomach was distended. It was noted on the CT. We don't know if this is a PSCA on-target effect. It wasn't particularly symptomatic, and we haven't seen it in other patients, but it's something we'll be looking at moving forward. His cytokine profile shows a strong spike in IL-6, as well as interferon gamma and a couple others that corresponded with the onset of his cytokine release symptoms. And when we look at the first 6 patients on study, the top 3 did not receive the lymphodepletion, and the bottom 3 did. So what we see is lymphodepletion really does seem to increase the amount of change in cytokines, since those bottom 3 graphs show much more movement compared to the upper 3 graphs, again, mechanistically supporting that lymphodepletion is important for response here. In terms of the persistence of the CAR T cells in the circulating blood, again, the role of lymphodepletion seems clear, with the black and gray lines without lymphodepletion on the bottom not showing much persistence over the month, whereas the blue and red lines are the patients who did receive lymphodepletion, and we see many more CAR T cells remaining in the circulation over the course of the month. So our next steps for developing this promising treatment do include a collaboration with Owen Whitty at UCLA as part of a SPORE that Dr. Preisman is working on. And the stains for CCAM and NCAM will be important there. We are seeing clinically some neuroendocrine dedifferentiation as patients progress off the study. We even had one who had that in his on-study biopsy. So we know this happens just with advanced prostate cancer that's heavily pretreated. But it's quite possible we're going to need a dual targeting of both the neuroendocrine and the more garden variety prostate cancer populations in order to achieve the kinds of durable complete remissions we're seeking. We're also going to be looking for the pre and post tissue samples to show us whether our CAR T gets to the tumor and gets exhausted, in which case adding an immune checkpoint inhibitor might be helpful, or whether it's having trouble penetrating into the tumor environment, in which case we might need more of a pull stimulus to achieve the kind of success we're looking for. Dr. Priceman is also working on single-cell RNA-seq, which will help look at the CAR T cells and also provide the potential to study for antigen spread. And as I mentioned before, we're looking at PSA staining, not only in the tissue, but also in the circulating tumor cells and the marrow rare cell populations to see how that evolves over time. We may consider multiple doses. This is something they're doing in other solid tumors, and our kinetics will inform that in terms of how frequently we might need to repeat dosing, how many doses, and finally, we may expand imaging to include potentially PET scans. So I'd like to really thank my scientific collaborators. Saul Priceman leads the T-cell therapy lab with many people working. We send them not only the specified study samples, but if we have a toxicity and we get a biopsy, we're sending that to them as well. So we're going to learn so much from every patient. Also, the Kuhn lab with this great collaboration, all the clinical and regulatory staff that are working on this protocol, and again, the Prostate Cancer Foundation for the funding that has led to the launch of this phase 1 trial. Steve Foreman has been a fantastic mentor, and it's really been great to collaborate with the whole CAR T team at City of Hope. And of course, we couldn't do this without the patients. We're doing this for the patients, but they are so committed, so brave, and just want to take a moment to recognize their role in the development of this exciting new therapy. Thank you.
Hello, I'd like to thank the organizers of the Prostate Cancer Foundation for inviting me to give this presentation of our novel TGF-beta receptor dominant negative to enhance prostate-specific membrane antigen chimeric receptor therapy in the treatment of patients with metastatic prostate cancer. Adoptive immunotherapy with CAR T cells is a novel approach for the treatment of prostate cancer. However, the prostate cancer immunosuppressive microenvironment has high levels of TGF-beta, which can limit the effectiveness of redirected T cells upon tumor infiltration. Therefore, we can use a dominant negative TGF-beta receptor to inhibit T cells and enhance tumor immunity. In the next few slides, I will show that in vivo disseminated tumor models, co-expression of the TGF-beta receptor dominant negative on PSMA redirected CAR T cells leads to increased T cell proliferation, as well as enhanced cytokine secretion, long-term persistence, and greater tumor eradication. So this is the first slide, which depicts the preclinical work from the June lab, which shows in the upper right-hand corner of the slide that T cells expressing anti-PSMA, specifically lyse PSMA positive PC3 cells. And in the right lower quadrant of the slide, you can see that expression of the anti-PSMA with the dominant negative enhances antigen-specific proliferation of CAR T cells. In a mouse model, the anti-PSMA CAR T cells alone, which you can see in the second row of mice there, can reduce tumor burden. But when combined with the dominant negative, we get a increased effect. And this is demonstrated by bioluminescence imaging. Further, the dominant negative TGF-beta receptor contributes to a 7-fold expansion of PSMA CAR T cells in vivo, which you can see on the lower left-hand part of this slide. So based on this data, we conducted the first in-human phase 1 trial using this anti-PSMA TGF-beta receptor dominant negative CART, with the primary objective being safety, and the other objectives are shown below there, including prostate cancer working group 2, PSMA measurement, and a number of correlatives. The study design shown here has 4 cohorts. We initially started with a 1 to 3 times 10 to the 7th per meter squared number of cells, and that was cohort 1. Following a Bayesian design, the next cohort, without any toxicity seen, we proceeded to a second cohort, which was 1 to 3 times 10 to the 8th per meter squared cells. We did not see appreciable toxicity at this cohort, and so we proceeded to adding a lymphodepletion regimen of Cytoxan and fludarabine, similar to what's used in the CART leukemia trials, to precede the CART infusion by 4 days. Following treatment of 1 patient, we found it necessary to reduce the dose level of the CAR T cells to 1 to 3 times 10 to the 7th cells per meter squared. And this cohort minus 3A, we treated an additional 3 patients. And you can see the schema below of how often the patients were seen, and that patients had a biopsy to begin with and a biopsy at day 10. We did allow retreatment with lymphodepletion chemotherapy after 28 days of observation. The eligibility included patients had to have a biopsy and demonstrate 10% expression of PSMA in the tumor tissue that was biopsied. The other eligibility criteria were fairly standard, but we did require patients to have at least 1 prior therapy with a second-generation anti-androgen therapy or a 17-alpha lyase inhibitor. The demographics are shown of our 10 treated patients on this slide. All patients were Caucasian and of typical elderly age. The median number of prior therapies was 3, but with a range of 1 to 4. 100% of patients, as I said before, had expression, received a second generation of an AR inhibitor, and 40% of the patients received chemotherapy. So what did we learn from these 4 cohorts? Basically, what I will show you in the next few slides is that we know that the PCR expression of CAR T levels, the cytokine levels, and the PSA response, as well as the use of LD chemotherapy, was very important in efficacy, similar to the leukemia trials. In this slide are demonstrated the serious adverse events experienced by patients in these cohorts, with cohort 1, 2, 3, and -3 shown at the top of the screen with their dose levels. The area that's highlighted in yellow is cohort 3. We treated 1 patient at this dose level. Although patients in cohorts 2, 3, and -3 experienced grade 3 or higher cytokine release syndrome, only the patient in cohort 3 had substantial toxicity. Unfortunately, that patient developed a prolonged CRS followed by multi-organ failure and eventually died of sepsis, bringing us to examine the lower dose cohort. Here are the peripheral blood CAR T transgene kinetics by quantitative PCR. And what you can see on this slide, if you look at the color codes, is that basically cohort 3 and -3 had higher copy numbers. The graph to the left is the absolute numbers, and then it's also depicted to the right as a log fold increase. So you can see that the last two cohorts were the optimal cohorts. And in this slide, we show the cytokine expression by cohorts. So again, the cohort 1 and 2 are the ones that didn't receive LD chemotherapy, and cohorts 3 and -3 at the lower parts of the slide are the ones that did receive lymphodepletion. And you can see that the cytokine, each graph has its own scale that you note to the left, but you can see that the cytokine levels were highest in cohort 3 and also elevated, although not as high, in cohort -3. This is the waterfall plot of all patients treated thus far. And you can see that just about everybody in cohort 3 had a decrease in their PSA. The patient in -3, the patient in cohort 3 obviously had the deepest response. We're still analyzing the RECIST criteria and the imaging responses. So in conclusion, PSMA TGF-beta dominant negative CART is feasible and safe at the current dose levels. The responses do appear to be dose-dependent and enhanced with lymphodepletion. This trial is still accruing, and we are going to be accruing to other solid tumor types that have expression of PSMA as a part of the phase 1 analysis. We do think that serial dosing is likely to be safe and more effective. And we are pursuing nanostring T-cell sequencing, analysis of circulating tumor cells, further cytokine analysis, and CAR-T traffic analyses, but we won't be showing that in this presentation. And finally, there is a phase 1/2 multi-center industry trial using serial dosing, which is also ongoing. Thank you very much for this presentation, and I welcome any questions that you all have.
Thank you, Howard. I really want to thank the organizers for allowing us to present our work today. It's really an honor to present to the PCF Foundation. The title of my talk is Development of a First-in-Class Shared Neoantigen Vaccine for the Treatment of Prostate Cancer. Vaccines in the context of prostate cancer treatment are not news. There have been multiple attempts. However, clinical benefit has been an elusive target. Suffice to say that the phase 3 of PROSPECT really exemplifies some of those efforts, where very interesting and perhaps enticing phase 2 data really did not succeed into showing patient benefit in phase 3. What we decided to do a few years back was to try to redefine how to train immune system cells to recognize prostate cancer antigens. And for that, I'm going to show you our efforts on the identification of new targets that are prostate cancer specific and that are able to elicit strong immune responses in the context of a vaccination strategy. The concept that I'm going to try to introduce to you today is that we can create a first-in-class, off-the-shelf, shared neoantigen vaccine that is based on a collection of neoantigens that we're going to call neo-ORs. And these neoantigens are prostate cancer specific, they're highly recurrent, and they're immunogenic. That in the context of optimized vaccination delivery vectors, in this case, viral vectors, adenovirus, as well as vaccinia vectors, and enhanced immune regimens, meaning checkpoint inhibition, will provide for a regimen that we think are going to be disease changing. So what do I mean with respect to neoantigens? I'm going to define neoantigens for this talk as a series of RNAs that are the product of transcription errors that result in the incorporation of coding sequences that lead to novel amino acid sequences. These incorporation of novel sequences are summarized in this picture. We're talking about novel introns, cryptic exons, out-of-frame fusions, and events that lead to series of amino acids that are incorporated in the context of a novel sequence that we are going to test their immunogenicity and the frequency at which it occurs in cohorts of cancer patients. Now, the key to this analysis is that some of these RNA species do not exist at the DNA level. They cannot be easily predicted. So they have to be found by doing extensive analysis of RNA databases. We obviously do look at other transcriptional errors that have been looked at in the past, including indels and single nucleotide variations. The key identity of the neoantigens that we're talking about is that they are shared neoantigens. That means that the same amino acid sequence is present in a significant fraction of the patients. That same amino acid sequence should not be present neither in control tissues or in other proteins, and they must contain enough amino acid sequence to be able to drive a new antigenicity. So what does this pipeline look like? We analyzed about 550 prostate cancer patient samples, both from the RNA dataset and DNA datasets. This included TCGA dataset, standard to cancer, as well as a number of control tissues represented in the GTEx databases. This is an example of the selection criteria, which contain a number of different parameters. One of the key parameters is that each one of the antigens has to be present in at least 10% of the patients. It has to be upregulated with respect to control cohorts. It has to be not present in normal tissues, and it must contain a sequence that is novel not only for the gene, but also for the genome. An example of outputs for these events are that we have a number of neoantigens that are really completely novel amino acid sequences, some of them coming from these intron retention events, alternative splice forms, as well as in-frame fusions. Now, with the in silico analysis, now we have a number of antigens that we plug into the next part of the pipeline, which is to understand if those antigens are really good candidates for a vaccination strategy. To summarize a lot of work, on the left-hand side, you see a panel that has colors. Every color represents a positive signal. We took all the neoantigens that were predicted from the TCGA dataset, and then we tested their expression on a completely new validation set. So on the y-axis, you see the novel prostate cancer samples, and on the x-axis, you see a number of different neoantigens that we predicted. As you can see in the red box, many of the neoantigens appear to be at very high frequencies present in the validation set. And not only that, but the colors really represent the strength of the RNA signals with respect to the presence of the neoantigen species. A lot of questions are related to the validation of those RNA species. Do they really exist as a full coding RNA? And the answer is that, to the best of our analysis, we have data that confirms about 85% of the predicted neoantigens for the pipeline. In addition to that, we continue to increase the number of protein level validation that we have found for these neoantigens. How many neoantigens you need to really build a vaccine that has therapeutic potential? You can see here that as we accumulate number of neoantigens, the patient coverage increases up to a plateau, with somewhere in the order of higher than 50 antigens. We also have to understand that not all the antigens have epitopes for all the HLAs. So once we factor in HLA configuration as well as possible precursor frequency, we really have to have a significant number of antigens present in the vaccine to be able to cover as many patients as possible. And What you see here is that we would like to have as many as 30 or 40 antigens in a vaccination strategy to be able to be successful in this enterprise. What I'm going to show you here is really our validation attempts to really understand if these novel sequences that we predict to be very immunogenic are really immunogenic. The first plot here on the left is really the control. Here we have immune stimulation of peripheral blood T cells with peptides coming from the neoantigens. The second plot is the positive control, which is the responses of those T cells with respect to known viral antigens. And then you see on the NeoORF 1, 4, 17, and 20 that we are able to induce very significant immune responses against neoantigens in vitro, meaning that there are a significant number of T cell precursors that can recognize these antigens. Indeed, every donor that we have tested have been able to have responses to neoantigens that we have tested. Now, to be able to build a successful vaccination strategy, we also have to focus on the vectors that are used to induce the immune responses. There is a lot of understanding now with respect to what vectors constitute good adjuvants for the production of robust immune responses in humans. In this case, we have decided to use a combination of adenoviral vectors and vaccinia vectors, plus likely an immune-enhancing checkpoint inhibitor. These combination strategies with viral vectors, have been successfully used in humans in the past. Indeed, this is one of our internal efforts in a non-human primate study where we have shown that our J&J-owned adenoviral platforms can really have antigen-specific CD8 responses against cargo antigens that can be significantly enhanced by checkpoint inhibitors. On the left-hand side, I just want to remind you that our neoantigens not only are present in a significant number of patients, indeed about 50% of the patients have around 15 NeoORFs or more. And that, if we break it down into the sequence of the NeoORFs, the HLA of the patients, and the putative antigens epitopes that are included in H neoantigen, in the middle picture, you see that our predictions have shown that about 50% of the patients contain at least 20 putative antigens that are able to match their own patient's HLAs. Indeed, I hope I have shown you enough information that now this neoantigen characterization can be really a new tumor vulnerability. And this tumor vulnerability would be founded on the findings of gene fusions, alternative splice forms, and point mutations that are really the new events that the immune system can use to really track down the cancer cells and elicit a potent immune response. We're definitely excited about taking this into the clinic. We would like to take this into an early stage prostate cancer, ideally biochemical recurrence with evidence of rising PSA, where the immune responses can be generated with enough time to be generated, and where we can follow PSA changes in patients where the disease may be able to be controlled by a vaccination strategy. We are very excited also about the possibility of use this heterologous prime boost in addition to multiple boosts, as the immune responses can be enhanced later on in patients with a boost or a CPI in addition to the treatment options. So just to end, this is really a major effort from Janssen Oncology. I'm not going to go through the details, but there are many departments that have been collaborating very extensively to be able to get to deliver this to our patients. Thank you so much for your attention. Happy to answer any questions.
Greetings and welcome to our Q&A session for that awesome set of talks on CAR T cells and novel neoepitope vaccines. We have Doctors Dorf, Haas and Sepulveda with us and my science partner, Dr. Miyahira. So it's just a real pleasure to be live and interact for a little bit and ask some questions. So I will take the first question and then I'll turn it over to Andrea. But my first question is for you, Dr. Dorff. PSCA has been a molecule that we've been interested in for many, many years. If Dr. Ryder is on the call today, this was one of his big projects when he was a fellow at UCLA. So we know a lot about it, but it's great to see it resurging as a targeting molecule for a CAR T cell. We're very, very pleased with the results to date. I'm wondering if you could tell us a little bit about the expression of this molecule throughout the history, natural history of advanced prostate cancer.
That's a great question. I think we still have a fair amount to learn, but at least I can say what we've seen based on the biopsy samples in our patients on the study. It appears to be very strongly expressed in most patients, even after multiple lines of therapy. And I'd really like to thank Dr. Ryder for the discovery and for his continued collaboration with the antibody that's allowing us to really interrogate that both in our tissue samples and in some other unique ways moving forward, including with the CUNE collaboration.
Thank you for that. I do not hear Andrea.
Oh, here I am. Sorry about that. Our next question is for Dr. Haas. The question is from Dr. Siddiqui at MD Anderson, and the question is, for patients that did not respond, what resistance mechanisms do you think are at play? And could it be due to inadequate T cell trafficking or contribution of the myeloid population?
So thank you for that question. We're working on those answers right now with some nanostring sequencing that we have been doing. I can tell you that we are seeing some of the markers of resistance that I think Dr. Reddick mentioned in his talk earlier with the PSMA BiTE therapy. And we think that there are probably some ways to get around it. But one of those ways may be to combine other targets with the PSMA TGF-beta.
Thank you for that. Dr. Sepulveda, first of all, thank you very much for making this the first public disclosure of the Janssen neoepitope vaccine. We're so pleased that you chose to present that at our meeting. So thank you and your colleagues very much. There's a question from Francesco Crea from the Open University in the United Kingdom. And this individual asks, do you think that some of your, quote, shared NA are also shared between adenocarcinoma and neuroendocrine prostate cancer variants?
So first, it's a privilege to present here. We have been friends of PCF for quite many years at J&J. The question is very interesting, and it really relies on current experiments that we're trying to undertake because we need access to the right databases. And indeed, maybe this is the perfect platform. If people have access to those databases, just try to get in contact with us. We are really excited about applying the bioinformatic tools that were generated at J&J that discover this new class of neoantigens to really ask those questions in as many samples as we can. There is a quality question that is incredibly important to be able to find this new class of antigens that has to do with the length of the RNA reads as well as the depth of the sequencing. This is really unique opportunity to thank really the bioinformatic team, which is really the driver within J&J on all of these discoveries. Thank you.
Okay. Let me just ask one more question to you, Dr. Sepulveda, that just came in from a medical oncologist in Melbourne, Australia, Shanine Sandhu. Shanine asks, is there consideration given about possible pruning of neoantigens in the setting of disease progression?
Yes, we have, with the databases that we have analyzed, which is roughly about 600 patient data points, we do know that progression of disease seems to be linked to a difference in what we call the universe of neoantigens, either because of the progression of disease itself or because of pressure by SOC. We don't know the answer to that, but we do see differences. We see that some neoantigens that are associated with the AR pathway are very highly expressed at early stages, and they change over time. That's also a question that we're trying to follow up on.
Thank you. I have a question, and I think is for both Dr. Dorff and Dr. Haas. So maybe Dr. Dorff, you can answer it first. How are you all thinking of addressing the tumor microenvironment in combination with your CAR T, or I guess this is for everyone, vaccination strategies, the CAFs, the cancer-associated fibroblasts, MDSCs, et cetera, are still present in the lesion and could limit efficacy? And this question is from Amy Moran at Oregon Science Health University.
That's the million-dollar question. We'll, of course, learn, as I mentioned, through the biopsies to see whether, in fact, there is an issue getting into the tumor microenvironment. But I believe MDSCs are a formidable barrier to success for immunotherapy in prostate cancer. And so there are specific therapeutics that are emerging to help deal with that. And I'm very interested to hear also what Dr. Haas has to say. But some strategies include targeting STAT3, combinations with checkpoint inhibitors, or potentially radiation, although I think there's more mixed literature on whether that's going to create the hot microenvironment that we want and overcome the things like the MDSC.
Dr. Haas, did you want to?
So I would equal what Tanya said, but I would also add that I think that we need to look at these CARTs earlier, with earlier stages of prostate cancer, perhaps the locally advanced population. Certainly tumor burden, whether it's in the tumor microenvironment in the bone or elsewhere, are significant hurdles. But I think in addition to what she mentioned, looking at it at perhaps the locally advanced population might also be interesting.
Mm-hmm. And Dr. Haas, I would add to that the construction, when you and Carl and the team produced that CAR T cell, the TGF-beta receptor dominant negative signal was indeed to overcome an immunosuppressive microenvironment. So, I think that was a very intelligent thing to do. Okay, I have Dr. Sepulveda. There were 3 quick questions that came in about your vaccine. So I'm going to give them to you one at a time, and if you can just give us a crisp, quick answer, it would be greatly appreciated. So the first one is from Lee Ellis from Cedars-Sinai now. Lee would like to know if you think that double-stranded RNA contributes to neoantigen discovery.
We do not have an answer for that. We have tried to look for that. It's not really clear.
Okay. The second one is from Christina Jameson, Chris Jameson at UC San Diego. And first of all, she says, really inspiring work with an exclamation point. Dr. Sepulveda, did you see any evidence of increased A to I RNA editing as a mechanism to produce neoantigens?
We see an increase in mechanisms that trigger alternative splicing. We do think that that's linked to some machinery linked directly to alternative splice events more than any other RNA modification pathways.
And then the third one from Jeff Smith is, do the neoantigens tend to occur in genes characterized by an increased rate of alternative splicing?
We think so. That's our current hypothesis, as well as genes that are highly expressed and genes that are linked to AR pathway.
Thank you very much for that.
Thank you, Dr. Sepulveda. I have the next question for Dr. Dorff. It's from Arpit Rao, and he asks, Why is lymphodepletion required in your opinion? And is it consistent with emerging data for PSMA CAR T? So maybe Dr. Dorff, you can comment on the role of lymphodepletion with your CAR T, and then Dr. Haas can give us her opinion also.
You know, I was skeptical as well for that solid tumors would require lymphodepletion, but it's pretty clear from Dr. Priceman's work in the lab that it improves persistence, which is necessary for ongoing, more durable remissions. And we think it impacts the tumor microenvironment in favorable ways, as well as impacting the circulating immune profile in a way that just helps the CAR T, when we put it in, do what it needs to do, go where it needs to go, expand and persist.
Okay, I have a question that we will address to both Doctors Haas and Dorff. This is coming from an investigator at MD Anderson Cancer Center, and it reads, for those patients that did not respond, what possible resistance mechanisms could be at play? Could it be due to inadequate T-cell trafficking, myeloid population? And I will use the bully pulpit to add target antigen loss as occurs in CD19 and some other CAR T cells. So I'd like your, whether it's preclinical, maybe too early to have clinical data on resistance, but what do you think is going on? Dr. Haas?
So I would answer yes to all of the first part of that question. Certainly what we're seeing when, so just to qualify, remember that in both of these trials, this is a feasibility. And so we first, in ours, we looked at a dose level, first a very low dose level that we thought was pretty safe. The second dose level, again, higher dose of CART, but no lymphodepletion. And then the third dose is when we added the lymphodepletion. And certainly when we're looking at the profiles of expected cytokines and things that we, that go up, we're seeing a much bigger kick with the lymphodepletion. And those are the patients where it's easier also to see what's happening with markers of resistance. And the way we're measuring those are with quantitative PCR for trafficking, but also with a biopsy, usually at 10 days or thereabouts. And from that, we're able to compare the nanostring. At least in some of the patients, we've done quite well with bone biopsies before and after the CART. And we are seeing, I think, evidence that that all of the things that were mentioned are things that we're going to have to figure out ways around. And as Howard was mentioning a few minutes ago, one of the other ways is to work on T-cell persistence, perhaps with things that you can turn on and off even as the CART is in the body. You can add switches to incorporate other mechanisms to deal with resistance. But this is, these are both very small trials. Our patients, we've treated 10 patients so far, so it's still really early days.
So another question for,
and I would add that
Please go ahead, Tanya.
Go ahead, Tanya.
Sorry. I would add that I really think the neuroendocrine population is an important part of the resistance story, and that dual targeting might be an important way forward. And then potentially repeated dosing. As I mentioned, that's a strategy that's being used in some other solid tumors if you just can't get enough expansion and persistence. So if you see a partial response and then a small progression event.
Okay, I think, Andrea, we are at the end of our time. I want to thank these three awesome investigators for their work that they did for us in the labs and the clinics leading up to these presentations. I wish we could all be together in Carlsbad/La Costa. Maybe next year, we're hopeful. But I hope that you and our viewing audience today have found this to be a useful format. We certainly have, and we're most grateful to all of you for being with us. Thank you.